Welcome back, guys. So what we're going to do now is we're going to use this slide to recap together the organelles or the pieces of puzzle that is going to create a cell itself. Okay, so can anyone tell me what is this big thing in the middle of a cell? If you answered nucleus, you would be correct. So this is a nucleus. Very good. Okay, how about a little bit more of a tricky one? What is this? This sort of flattened looking membrane thing that seems to have little spots on it. What is that? Well, if you said ER, you would be absolutely correct, but you also need another part of that ER, and that is rough ER, okay? Why is it rough? Because it has these spots. Now, who remembers what these spots are? If you said a ribosome, you would be absolutely correct. Ribosomes, remember, are the ones that are that can be in two different structures. They can be freely floating in the cytosol, and that's these little guys here, okay? Or they can be attached to the ER membrane, which means that that part of the ER is rough ER, and the little spots on them are ribosomes. Okay, what's the function of all of these things? Nucleus itself contains something we called DNA. And we now know that DNA is genetic material of your cells and of your cells. And we now know that the DNA itself can be in this sort of stringy long mode, or it can be in condensed form, which is called chromosomes. Okay, all of that is inside of the nucleus, and on the outside of the nucleus you have a nuclear membrane that's perforated or has little pores called nuclear pores. I'm just writing NP for nuclear pore. Okay. All right. What about the function of ribosomes and the ER? What is the function of the rough ER and the ribosomes? If you said protein synthesis, you would be absolutely correct. So these two guys together make proteins. Okay, and we'll talk about that particular process in the next session. Good. That's excellent. All right. What about this guy here? What is that? Does anyone have any idea of what this is? If you said smooth ER, you would be absolutely correct. Smooth ER is the ER that doesn't contain any ribosomes, and it's a point of lipid synthesis. Okay. What was that one thing that I also wanted you to know about the ER? Do you remember? Something that will be important for later sessions? Yes, that's it. Calcium storage. Okay. Very, very important for you to remember that. Okay, good. Now, if you make proteins here in the rough ER using the ribosomes, and these proteins are not folded properly, what happens to them? They get that death stamp, remember, called the ubiquitin, and the ubiquitin proteins go into this structure here. Does anyone know what this structure is? What's it called? Proteasome. Absolutely correct. Proteasome. Remember, that's that barrely looking big organelle where ubiquitinated protein comes in on one end, gets digested through, and the proteasome is able to release the protein fragments or peptides. Very good. Nice. All right. What about this funny looking thing? What is that? Well, if you said mitochondria, you would be absolutely correct. Mitochondria is the site of what? What happens in the mitochondria? Correct. Mitochondria is the site of ATP synthesis, and it's the site of ATP synthesis because it has this inner membrane, that crista, that folded inner membrane that is studded with all these enzymes that make this ATP. Great job. All right, what about this one? This big one. What's this called? Golgi, you reckon? You're absolutely correct. Golgi is what we called a bus stop, right? Or a sorting platform. It's a site where proteins that are made in the ER are packaged into and given a little stamp or ticket to which bus to board. Okay, that's really good. So the Golgi, the site of post-translational modification. Post-translational modification being the addition, for example, of a sugar molecule on a protein or a lipid molecule on protein, which serves as a ticket for that protein to know where to go inside the cell. So sometimes it goes outside of the cell, sometimes it stays in the plasma membrane, sometimes it goes back to a different organelle. Excellent. Now, what about this funny looking thing here? What is that? Centromere? Correct. What is the centromere made of? Exactly. Centromere is made out of microtubules, and microtubules is a type of cytoskeleton. You're precisely right. Now in this picture, you really don't have the cytoskeleton represented in a very ordered form, but we now know that there's 3 different types of cytoskeleton: intermediate filaments, microfilaments, and microtubules. And all 3 of them, they do the support of the cell, but they also have different functions because they are composed of different proteins. Excellent. All right. What about this? What do you think this is? Cilia, you reckon? You would be correct. Cilia are these extensions of the plasma membrane that are supported by these lines, and these lines are microtubules, right? So protrusions of microtubules covered by the plasma membrane is what the cilia are. The other part of the protrusion of the plasma membrane is also flagellum. Okay, flagellum, you only have 1 flagellum per cell, and you only find that in the sperm cells. Okay, good. This guy here, what does that one represent? I'll leave this one up with you. Have a look in your textbooks. Have a look back at the sessions, and try and answer on the loop what is that particular structure. Great job. Okay, so we've recapped the session itself. Now you know the little puzzle pieces, both the structure and function of all the puzzle pieces. From next session, we're going to start putting these puzzle pieces into big functions. So we're going to learn how to make a protein. We're going to learn how the cell communicates with outside world. And we're also going to learn how the cells take things in and spit things out, okay? Now, in order for you to keep going and understand the next session, please complete the concept map for session 1. You have your concepts on the map 1, see map 1 document. For next session, before the next session, can I please ask you to make the model of the plasma membrane? You will find all the instructions in the session 2 tutorial. If you make this, it will be easier for you to understand certain things. Also in the same tutorial, you will find the osmosis experiment, and I would like you to put on the loop what is it that you received from that experiment. So what are the actual outcomes of that experiment, so that we can put it into a particular part of the next session that we talk about, when we talk about a function called osmosis. So that's it. That's your session 1 done. Hope you enjoyed it, and I will see you in the next session. Bye.